After class on Thursday, I was asked if I could explain this problem with a short video. So I'm going to take a few minutes to go over each of these parts of this problem. So the problem starts off here with, in cats, curled ears result from an allele big C big U that is dominant over an allele little c u for normal ears. And then the coat color is coded by this gene G, where big G codes for the color black and little g codes for the color gray. You also had a problem similar to this in your Achieve homework. And in your Achieve homework, they use the capital CU and the little cu here. I think this causes some confusion because you're using two letters. And so as I explain this problem, I'm just going to use capital C and lowercase c and drop the U part of it just for simplicity. So let's tackle the first question here. When I have a problem like this, I like to write down everything I know first and then see how I'm going to use it. So I'm going to say big C, and big C, little c equals curled ears, and little c equals normal ears. Big G equals black fur, and little g equals gray fur. And to be consistent, I should say big G, and big G, little g equals black fur. So they say in this problem, a gray cat homozygous for curled ears is mated with a homozygous black cat with normal ears. What are the genotypes of these parents? So let's start with the gray cat. In order for the cat to be gray, the only way the cat can be gray is if they are little g. That's the only way you can have the gray coat color. And then as far as curled ears is concerned, the problem tells us that they are homozygous for curled ears. So we know they have to be big C. Now for the other parent, we know that they are homozygous black. And so that means they're big G, right here. Because a black cat can either be big G or big G, little g. But the problem tells us they're homozygous, so we know they're big G. And then they say they have normal ears. So if they have normal ears, we know the only option is little c. Okay, so that's the answer to the first part. And I'm going to copy this because we're going to use it for the next question. So we're going to take the parents from the previous question, little g, big C times big G, little c. These are the parental cross. And when you make them together, you generate the F1. And we know all the F1s have to be big G, big C, little c. So let's work out over here why we know that they have to be heterozygous for both of these alleles. We're going to just do two simple monohybrid crosses, little g, big G. And we know this parent is only going to be able to give little g, and this parent here is only going to be able to give big G. So we know all of the progeny have to be big G, little g, because that's the only thing the two parents can give them. Same is true when we look at this second set of alleles. This parent can only give a big C allele, and this parent can only give a little c allele to their progeny. So we know that the progeny have to be big C, little c. So we know that the genotypes of the F1 have to be big G, big C, little c. So the next part of this problem is going to work with these F1 progeny. It says two F1 cats are mated, and these were the F1 cat genotypes. They're mated together to generate F2s. And how I would solve this is using the fork branched method. So big G times big G, little g. We know 3/4 of these will be big G, and 1/4 will be little g. Now let's look at the C gene. We know that if you take two heterozygotes, big C times big C, that 3/4 of them will be big C, or big C, little c. And I should correct that down here. This is 3/4 that are big G, or big G, little g. And we know 1/4 of them will be little c, little c. Now this combination is just as likely to pair up with the big G phenotype, the black phenotype, as it is with the gray phenotype. So we have to repeat this down here to combine with the little g, little g. So we'll multiply 3/4 times 3/4, and I'll show you that number in a moment, and also times 1/4. And then we need to multiply this 1/4 g, little g, times this combination here. And then we'll see what this is equal to in just a moment. So we know 3/4 times 3/4, that is 9/16, and that means 9/16 of them will be big G, big C phenotype, or we could say black with curled ears. We're not saying for sure what that second allele is, but we know it has to be either big G or little g. But it doesn't really matter because the phenotype is what we're curious about. Because that's what the problem is. What are the phenotypes of the F2? Then we multiply 3/4 times 1/4 and we get 3/16 big G, little c, or we could say black with normal ears. Then down here, it is 1/4 times 3/4, so that's going to be 3/16 little g, and big C. And again, we don't know if that's big C, or big C, little c, but it doesn't really matter because what the question is asking for is the phenotype. And so we know this phenotype would be gray with curled ears. And then our last phenotype is going to be 1/4 times 1/4, that's 1/16 little g, little c. Or phenotypically, what we would say is gray with normal ears. In fact, you may recognize this 9:3 to 3:1 ratio as a very common ratio. The next question, the last question, it says do it on your own, but we'll do it here. We'll just get rid of that then. An F1 cat mates with a stray cat that is gray. So again, remember our F1 cat was big C, big G, little g. And it mates with a gray cat that has normal ears. So normal ears tells us they will be little c, and gray we know is going to be little g. What phenotypes and proportions of progeny are expected from this cross? So again, we're going to use a forked branch method to solve this problem. First, if you take a big C times a little c, you know that 1/2 of them will be big C phenotype, or have curled ears, right? And 1/2 of them will be little c phenotype, that is the normal ears. And to show you how we know that, it's just that simple Punnett square we could do over here, where you do big C, little c, and you just combine the alleles. And with lowercase c, I often just put an underline mark under it because sometimes the size of the c is arbitrary. And so you can see then, when you do this Punnett square, that you have 50% are big C, and 50% are little c. So 50% have curled ears and 50% don't. Now we need to combine it with the G alleles. Just like we did on the previous slide, we know that 50% of these will be big G phenotype, that is black, and 50% will be little g phenotype, which is gray. You can do a Punnett square just like I did over here, but it's the same concept. And just as we saw in the previous slide, the G alleles are just as likely to combine with big C's versus little c's. So we know again, half big G, half little g. And here and again, what you do is you just multiply this through, like we did on the previous slide. So half big C times half big G, you get 1/4 big C, big G, or we could say curled ears with a black coat. We multiply the second row, 1/2 C, 1/2 little g, and you get 1/4 big C, little g, or curled ears with a gray coat. We do the same on the bottom half here, 1/2 times 1/2, we get 1/4 that are little c, big G, or we could say normal ears with black coat. And the bottom one here, we go 1/2 times 1/2, we get 1/4 little c, little g, little g. Or we could say normal ears with gray coat. So we end up with a 1:1 to 1 to 1 ratio here. Now I noticed when I did this one, I put the big C first, that is the gene that codes for ear shape, and the color of the fur second. On the previous problem, I did it reverse. I put coat color first and ear shape next. I didn't do that for any particular reason. It's just the way it worked out. But it doesn't really matter which one you do first or which one you do second. Okay, that was the last of this problem. If you have any questions, let me know, and I hope this was helpful. I'll see you in class. Bye.